Welcome to The Deep Dive, the show where we take a stack of fascinating sources and distill them into the most important nuggets of knowledge just for you. Today we're plunging into a world that's entirely unseen, a universe thriving right beneath our noses, constantly impacting us in profound ways, from the diseases that challenge us to the very process of our digestion. How much do we really know about the microscopic inhabitants of our planet, specifically bacteria? Get ready, because we're about to peel back the layers on these tiny powerhouses.
It really is an incredible realm, and our deep dive today is into general bacteriology and the intricate morphology and physiology of bacteria. You've shared some incredibly detailed sources, really comprehensive stuff that gives us a great look at these fascinating microorganisms.
Yeah, exactly. Our mission for this deep dive is clear. We're going to take these rich sources, and they cover everything, right? Foundational medical microbiology, historical breakthroughs, detailed breakdowns of bacterial structures, their unique life processes, how they interact with their environments, and we're going to extract the most vital knowledge and insight. Think of it as your shortcut to truly being well-informed about the microscopic architects that shape our world. We'll uncover some surprising facts, hopefully get to those aha moments that make understanding this invisible realm truly captivating. It really is a treasure trove of information. It's going to reveal the immense complexity hidden within the microscopic. So to really understand this invisible world, we probably need to start with the very definition. When we talk about medical microbiology, what exactly are we talking about?
Right. So medical microbiology is the branch of science that specifically deals with the causative agents of infectious diseases in humans. That's the core of it. It explores how humans react to these infections, the specific ways these agents produce disease, and really critically, the methods used for their diagnosis. But it's also a surprisingly vast field. It actually encompasses much more than just bacteria.
Oh, okay. So it's broader than our focus today, then.
Oh, yeah, absolutely. While bacteriology is our core focus, medical microbiology also includes things like general microbiology, immunology, which is fascinating, studying how our bodies fight back against these invaders, virology, which is viruses, mycology, the study of fungi, and parasitology. What's truly interesting, though, is how interconnected these fields are. I mean, understanding one aspect, say, the structure of bacterial cell wall, that often illuminates how our immune system recognizes it, or how we might develop a new drug to target it. They all kind of build on each other to give us this holistic understanding of health and disease and, ultimately our well-being.
And for you, the listener, it's crucial to grasp this foundational definition because, well, it really sets the stage for appreciating the sheer complexity and significance of microbial interactions with our bodies, with the broader environment. This isn't just abstract science. It directly impacts our health, our daily lives. Okay, now to truly appreciate where we are today in our understanding of bacteria, we have to go back, way back to the beginning. For centuries, these organisms were completely unknown. Who first lifted the veil on this invisible world? Who's the first person to actually see these tiny creatures?
Well, that credit really goes to Antony van Leeuwenhoek back in the 17th century. He was this incredibly curious, self-taught scientist. He painstakingly constructed the first microscopes that were powerful enough to reveal these, unseen entities. His groundbreaking achievement wasn't just building the device, though. He was the first person to actually observe microorganisms, which he called animalcules. Little animals, right?
But can you imagine seeing life where nobody had seen it before?
Exactly. Imagine the sheer wonder, and maybe, a little bit of terror, too, of suddenly seeing life where no one had before. His meticulous observations provided the first accurate description of bacteria. He essentially opened up an entirely new scientific field, a universe previously unimaginable to human perception.
That's truly incredible. So from just seeing these tiny forms of life, the next giant leap was figuring out what they were doing, especially in relation to disease. And this is where it gets really transformative, right? Enter Louis Pasteur. Pasteur didn't just observe, he fundamentally revolutionized our understanding of disease. How did he manage that?
Pasteur is monumental. Yeah, for several reasons. Before him, the prevailing belief was spontaneous generation. This idea that life could just spontaneously arise from non-living matter. Explaining things like maggots appearing in meat or microbes suddenly appearing in broth. Pasteur, through his really elegant swan neck flask experiments, he definitively disproved that theory. He showed that microorganisms don't just appear out of nowhere. They come from pre-existing microorganisms in the air or surroundings. And this was the birth of his revolutionary germ theory of disease, which basically stated that specific diseases are caused by specific microorganisms. A radical idea at the time.
And his practical applications are still critical today, aren't they? I mean, things we use every day.
Absolutely. His work wasn't just theoretical, not at all. He developed sterilization techniques which fundamentally changed practices in medicine and food preservation. Think about pasteurized milk. That's a direct legacy of his insights into killing harmful microbes with heat.
Right. Pasteurization.
Of course. And he also coined the very term vaccine, building on earlier work but really defining it. His pioneering work on vaccines for diseases like anthrax and rabies were monumental steps in preventative medicine. He truly brought about a paradigm shift for science and medicine. Before him, the true causes of many illnesses were just a mystery, which led to all sorts of ineffective, sometimes harmful treatments. His work provided a rational basis, a framework for understanding and combating infectious diseases, ultimately saving countless lives.
So building directly on Pasteur's insights, we then see Joseph Lister emerge. He's often called the father of modern surgery. How did he apply this revolutionary germ theory to, well, the gruesome reality of surgery in his time?
Lister made a direct and incredibly powerful application of Pasteur's germ theory right into the operating room. And frankly, operating rooms back then were often death traps. In the 19th century, post-operative infection rates were astronomically high. Infection often led to death. Surgeons didn't wash their hands between patients, instruments were reused without cleaning. The air itself was thought to carry miasma, but it was really full of unseen pathogens. Lister's major contribution was developing a system of antiseptic surgery. His revolutionary idea was to use carbolic acid phenol as an antiseptic. He used it to sterilize instruments, wounds, even surgeons' hands, and sometimes sprayed it in the air. This dramatically reduced infections and transformed surgery from this incredibly high-risk endeavor into a more life-saving procedure. It was a practical application of germ theory that had immediate impact.
And finally, in this lineup of pioneers, we come to Robert Koch, often called the father of bacteriology, for his really systematic approach to actually identifying specific pathogens. What were his key contributions? How did he move microbiology from just observation to rigorous science?
Yeah, Koch really transformed microbiology into a truly scientific discipline. He moved it from mere observation, which was amazing in itself, to controlled, reproducible experimentation. That was the key shift. He developed specific staining techniques that allowed scientists to visualize bacteria much more clearly under the microscope, and he developed the hanging drop method, which was really useful for observing bacterial motility, seeing if they could move. But perhaps his most foundational contribution, the real game changer, was devising methods for cultivating pure bacterial cultures.
Pure cultures. Why was that so critical?
Because before Koch, scientists were often dealing with mixtures of different microbes. It was impossible to definitively say which one caused what. Koch figured out how to grow bacteria on solid media, like agar derived from seaweed, which allowed him to isolate a single bacterial species. He could grow colonies, each starting from a single cell. This meant researchers could finally isolate a specific bacterium and study its effects in isolation. It allowed for true cause and effect research, linking one microbe to one disease.
And his discoveries themselves were profoundly impactful, identifying the causes of major diseases.
Oh, absolutely. He identified the causal agents of devastating diseases like anthrax, tuberculosis, TB, a huge killer then and now, and cholera, massive discoveries. His enduring legacy includes Koch's phenomenon, which describes a specific immune response related to TB, and most famously, Koch's postulates. These provided a rigorous, step-by-step framework for proving that a specific microbe caused a specific disease.
Okay, so we have these amazing discoveries, but how do we actually prove a microbe causes a disease? Koch didn't just find bacteria. He gave us this set of rules, his postulates, that fundamentally changed how we prove causation. How do these rules of the game actually work?
Right, Koch's postulates. They were formulated in the late 19th century, and they are still foundational guidelines. I mean, scientists use them today, even if they've been modified a bit with modern understanding. They provide this clear, logical framework for establishing that specific microbes cause specific diseases, especially bacteria. Let's walk through them conceptually. First, postulate 1 states, the organism must be shown to be invariably present in characteristic form and arrangement in diseased tissue.
Okay, invariably present. So basically, if someone's sick with disease X, microbe Y should always be found in their affected tissues.
Exactly. Not just sometimes, always. It's about consistency. You have to find the suspect microbe in every case of the disease you examine.
Okay, so if it's always there in the sick subject, what's the next step? You can't just stop there.
No, you can't, postulate 2 says. The organism, which from its relationship to the diseased tissue appears to be responsible for the disease, must be isolated and grown in pure culture. This means you take that microorganism you found in the diseased subject and you grow it completely alone in a laboratory setting, free from any other microbes. This was Koch's crucial innovation we just talked about, getting that pure sample.
Right, the pure culture. And once you have it isolated and pure, how do you really prove it's the culprit and not just, hanging around?
Then comes the critical test, postulate 3. The pure culture must be shown to induce the disease experimentally when inoculated into a healthy, susceptible subject. So you take that isolated pure microbe and you deliberately introduce it into a healthy, vulnerable host, usually an experimental animal back then. The expectation is that this healthy host should then develop the exact same disease with the same symptoms.
That sounds like the smoking gun, doesn't it? If the pure microbe causes the same disease again.
It's a huge step, yes. But to be absolutely certain, to really close the loop, there's postulate 4. Postulate 4 completes the cycle. The organism should be re-isolated from the experimentally infected subject and shown to be identical to the original causative agent. This means you should be able to find the exact same pathogen again in the newly diseased host, culture it again, and confirm it's identical to the one you started with. It proves the chain of causation.
Understanding the enduring value of these rules in medical science, even with all our modern advancements, is immense. They provide such a clear, logical framework for understanding and identifying the causes of infectious diseases. But it does raise an important question, doesn't it? I mean, in what scenarios might these postulates be challenging to meet today? And how do scientists adapt?
That's a really key point. They don't always apply perfectly, especially now. For instance, what about diseases caused by multiple pathogens working together? Or pathogens like certain viruses or bacteria that are just impossible to culture in a lab using current methods?
Right, or even asymptomatic carriers. People who have the microbe and can spread it but show no symptoms themselves. That breaks rule 1, doesn't it?
Exactly. And what about ethical considerations for postulate 3? We obviously can't intentionally infect humans with dangerous pathogens, so scientists have adapted. We use molecular methods now, looking for genetic fingerprints of microbes, using animal models carefully, studying immune responses. Our understanding has definitely evolved, but the core logical principles laid out by Koch remain incredibly influential.
Okay, let's step away from history for a moment and talk about the big picture of life itself. You mentioned earlier that all living organisms fall into one of 2 fundamental categories, prokaryotes and eukaryotes. What makes them so fundamentally different from each other, and crucially from us humans?
Right, this is a truly fundamental divide in biology. Understanding it dictates so much about how bacteria operate and how we interact with them, especially with things like medicine. So, eukaryotic cells, that includes all animal cells, plant cells, fungi, protists, basically almost everything we typically see around us, including ourselves, are defined by having a clearly defined nucleus. This nucleus houses their genetic material, their DNA, and they also have a host of other membrane-bound organelles like mitochondria, which are the powerhouses of our cells, or the endoplasmic reticulum for protein processing and so on. They're complex, compartmentalized factories.
Okay, so eukaryotes are complex with a nucleus and organelles. What about prokaryotes then? Where do bacteria fit?
Prokaryotic cells are much simpler, and they are typically unicellular organisms. They do not contain a membrane-bound nucleus or those complex organelles that eukaryotes have. Our focus today, bacteria, falls squarely into this prokaryotic category, as do another group called archaea, which often live in extreme environments.
So the nucleus is a major differentiator. That seems like a really big deal structurally. What about other key differences in structure or function?
The nucleus is absolutely central, yeah. In eukaryotes, it's present and membrane-bound, protecting the DNA. In prokaryotes, it's absent. Their genetic material, the chromosome, simply resides in a region called the nucleoid, which isn't enclosed by a membrane, it's just kind of there in the cytoplasm. Size is also drastically different. Eukaryotic cells are significantly larger, typically ranging from 10 to 100 micrometers across. Prokaryotic cells, in stark contrast, are much, much smaller, usually ranging from less than a micrometer to just about 5 micrometers.
Tiny. That size difference must impact everything else, right? Their internal complexity, how they interact with the world.
Absolutely. It directly impacts their internal complexity and how they interact with their environment. Think about surface area to volume ratio. Smaller cells have a much higher ratio, which is great for nutrient uptake and waste removal.
And how about their genetic material itself and how it's handled? That's obviously vital for life.
Right. DNA replication in eukaryotes is highly regulated, very precise, with selective starting points. And it's often a slower process involving multiple chromosomes, typically linear ones. Prokaryotes, however, usually replicate their entire genome, which is typically a single circular chromosome, much more rapidly. It's a simpler, faster process overall. And as for chromosomes, eukaryotes have more than one, organized linearly within the nucleus. Prokaryotes typically have that one long single loop of DNA, but they also often have additional smaller circular pieces of DNA called plasmids. And these plasmids are a really big deal for bacterial evolution and adaptation, especially things like antibiotic resistance. We'll definitely come back to those.
Okay, plasmids, got it. What about overall organism type? Multicellular versus single-celled?
Eukaryotes are usually multicellular, forming complex organisms like plants and animals, though some, like yeast or amoebas, are unicellular. Prokaryotes, on the other hand, are predominantly unicellular. Though sometimes they form colonies or biofilms, each cell remains fundamentally independent.
And even the cell's protein factories, the ribosomes, are different. You mentioned that earlier.
Yes, and this is a crucial distinction, especially for medicine. Eukaryotes have larger ribosomes in their cytosol, designated as 80S ribosomes based on their sedimentation rate. Prokaryotes have smaller 70S ribosomes composed of slightly different protein and RNA subunits, 50S and 30S. This difference might sound small, but it's incredibly important. It's the reason why certain antibiotics, like tetracyclines or macrolides, can selectively target bacterial protein synthesis and kill bacteria without significantly harming our own larger 80S ribosomes. It's a fundamental vulnerability that medicine exploits.
That's a fantastic example of how these fundamental biological differences translate directly into practical implications for us. What about their overall internal structure and growth rates, wrapping up this comparison?
Well, as we said, eukaryotes have complex organelles present within their cytoplasm, allowing for compartmentalization of different functions, energy production here, protein folding there. Prokaryotes lack these membrane-bound organelles, meaning all their metabolic processes occur directly within the cytoplasm or associated with the cell membrane. It's a more streamlined, less compartmentalized setup. Both cell types, of course, possess the fundamental ability to store hereditary information in their DNA. Lastly, the cell wall structure also differs significantly. Eukaryotic cells, if they have a cell wall like plant cells have cellulose, fungi have chitin, it's typically structurally simpler compared to bacteria. In prokaryotes, the cell wall is complex and present in almost all bacteria. It's primarily made of peptidoglycan, a unique molecule, and it provides crucial structural support, shape, and protection against osmotic stress. What's really fascinating here is how these fundamental differences dictate everything about their survival, from how they replicate at sometimes astonishing speeds to how we can target them with medicines. Their relative simplicity in many ways is actually their strength, allowing for rapid adaptation.
It's so easy to forget that for centuries, these microorganisms were completely invisible to us. Without microscopes, our understanding of disease, of life itself, would be drastically different. These instruments are absolutely essential for magnifying microorganisms that are far, far beyond the resolution limit of the naked eye.
Indeed. And microscopy has evolved dramatically since Leeuwenhoek's time. We now have a whole toolkit offering different windows into the microbial world. The most basic type is still the light microscope, which uses visible light passing through the specimen as its illumination source. It's used to study the morphology, the shape and structure of bacteria. We can look at them either in their living state, maybe swimming around in a wet mount, or more commonly after they've been fixed and stained to make them more visible.
And what about variations on that basic light microscope? Are there ways to see things better without staining sometimes?
Yes, there are. For instance, there's the dark ground microscope. This is a variation of light microscopy that cleverly illuminates the specimen from the side, so only the light scattered by the specimen enters the objective lens. This creates a dark background, or field, with only the specimen appearing brightly illuminated. It's particularly good for making unstained, living specimens easier to observe, especially thin ones like spirochetes. You see these bright outlines against a black canvas. Then there's the phase contrast microscope. This is a more sophisticated type of light microscope that takes advantage of the fact that light waves shift slightly as they pass through different parts of a cell. It enhances contrast by converting these subtle phase shifts into visible differences in brightness. Crucially, it does not require the use of staining procedures, which often kill the cells. This allows for observation of living, dynamic bacteria, which is vital for studying their movement, cell division, or other cellular processes in real time.
Okay, so those enhance what we can see with visible light. But then we get into specialized types that use different light sources or even entirely different principles, allowing us to see details we never could before.
Exactly. The fluorescent microscope, for example, uses fluorescence instead of just visible light as its illumination source. It illuminates the specimen with UV light or specific wavelengths of visible light. It usually requires specific fluorescent dyes like rhodamine or fluorescein, or even genetically engineered fluorescent proteins to bind to specific structures in the cells, making them glow brightly against a dark background. This allows scientists to highlight particular components like the cell wall or DNA or specific proteins, and even track specific molecules within a living cell, revealing specific functions or locations. And for the ultimate in magnification and resolution, allowing us to see incredibly fine details down to almost the molecular level, we have the electron microscope.
Right, the really powerful ones. They don't even use light, do they?
No, they illuminate specimens with a beam of electrons instead of light. Since electrons have a much shorter wavelength than visible light photons, electron microscopes can achieve much higher magnification and resolution. We're talking magnifications potentially hundreds of thousands of times, revealing details far beyond what any light microscope can see.
And there are two main types of electron microscopes, right? Each giving us a different kind of view.
That's correct. There's the scanning electron microscope, or SIM. This is used to visualize the surface topography and three-dimensional shape of specimens. The electron beam scans across the surface, and detectors pick up secondary electrons knocked off the surface. This gives us those stunning, detailed, almost sculptural images of bacterial surfaces showing their texture, appendages like flagella or pili, like tiny, intricate landscapes. Then there's the transmission electron microscope, or TEM. This is used to view ultra-thin sections of specimens. The electron beam passes through the specimen, and differences in electron density create the image. So TEM reveals the internal structures, the cell wall layers, the cytoplasm, ribosomes, the nucleoid region, almost like slicing through the bacterium to see what's inside at incredibly high resolution.
Wow. Imagine how different our understanding of bacteria and disease would be without these incredible technological advancements that literally allow us to see this unseen world. It's mind-boggling. So, okay, before we dive into the really nitty-gritty of their internal machinery, let's just cover some core characteristics that broadly define bacteria and how scientists actually go about studying their tiny details, given how small and often transparent they are.
Right, some defining features. At their core, bacteria are truly microscopic. As we said, typically ranging between 0.5 to 5 micrometers. Just to give you a sense, maybe 50 E. coli could fit across the diameter of a human hair. And it's worth noting, some of the smallest bacteria, like mycoplasmas, are even tinier, maybe around 300 nanometers in diameter, which actually starts to approach the size of the largest viruses. They really push the lower limits of cellular life. They are, as we've established, prokaryotic microbes, meaning no membrane-bound nucleus, no mitochondria, no other complex membrane-bound organelles, simpler internal organization.
And their genetic material is also quite distinct from ours, right? Not just the lack of a nucleus.
Yes. They generally possess a single supercoiled circular chromosome as their primary genetic material. It contains all their essential genes. And bacteria are haploid, meaning they have only a single copy of each gene. This differs from diploid organisms like humans, who have 2 copies. This haploid nature means mutations can have a more immediate effect, potentially speeding up adaptation. Another defining feature is that they are typically enclosed in a rigid cell wall, primarily made up of that unique molecule, peptidoglycan. This wall is absolutely crucial for maintaining their shape and protecting them from osmotic lysis, basically, bursting if the external environment is too watery. Some bacteria, in addition to their plasma membrane and cell wall, contain an additional extracellular layer called a tapsule or slime layer, which adds another layer of protection or helps them stick to surfaces. We'll get more into that. And generally, bacteria do not contain chlorophyll, the pigment used for photosynthesis in plants and algae. Though some specialized photosynthetic bacteria exist, they use different pigments and mechanisms.
Okay, so they're tiny, prokaryotic, have a unique cell wall, usually a single circular chromosome. But if they're so small and often colorless, how do scientists actually study their details in the lab? How do you see these things clearly?
Yeah, that's a fundamental challenge. Because bacteria are mostly colorless, staining becomes absolutely necessary for most light microscopy. Staining allows us to visualize them clearly, and importantly, to study their structure, shape, and other characteristics that help in identification. There are various methods. Sometimes unstained wet mounts are used, especially for observing living, motile bacteria. You just put a drop of liquid culture on a slide. Since you don't fix or stain them, you can see them moving around, which can be informative. but more often we use stained preparations. Simple staining involves using just a single basic dye like methylene blue or crystal violet, which colors all the bacterial cells uniformly, making their shape and arrangement visible. Then there's differential staining, which is much more informative. This uses multiple stains to differentiate between different types of bacteria or different structures. The classic example, the cornerstone of bacteriology, is gram staining, which divides most bacteria into gram-positive and gram-negative based on their cell wall structure. Acid-fast bacilli, AFB staining, is another differential stain, crucial for identifying bacteria like Mycobacterium tuberculosis, which have waxy cell walls. And finally, there's special staining techniques designed to highlight specific structures that don't stain well with other methods. For example, negative staining uses acidic dyes like India ink to stain the background, leaving structures like capsules visible as clear halos around the cells. Silver impregnation methods can be used to make very thin structures like flagella or spirochetes visible. And spore staining uses heat to drive stain into resistant endospores.
So these fundamental features define their existence and differentiate them from other life forms. And the challenge of their small size is precisely what led to the development of these critical visualization techniques, allowing us to even begin to understand this incredible invisible world. Now let's get into their appearance. Bacteria aren't just tiny uniform blobs, are they? They come in an incredible variety of shapes and arrangements, which can often tell us a lot about them just by looking. These distinct forms often provide immediate clues about how they might interact with their environment, adhere to surfaces, or even cause disease.
That's absolutely right. The morphology or shape of bacteria is a key classification characteristic, and it really does offer immediate diagnostic clues in many cases. The basic forms are spheres, which we call coccus, and rod-shaped cells, which we call bacilla, singular bacillus. Then there's a more complex group with spiral or curved shapes. Let's start with coccus. These are spherical or sometimes slightly ovoid cells, truly resembling tiny berries under the microscope. And importantly, they often arrange themselves in distinct patterns based on how they divide and whether they stay attached after division. These arrangements are often diagnostically relevant. For example, diplococci are arranged in pairs, often looking like two coccus flattened against each other. Streptococcus pneumonia, a common cause of pneumonia and meningitis, often appears this way. We also see streptococci, where the coccus divide along a single plane and remain attached, forming chains of varying lengths. A common example is Streptococcus pyogenes, the culprit behind strep throat and other infections. Think strep like a strip or chain. Then there are staphylococci. These divide in multiple random planes and remain attached, forming irregular clusters that truly resemble bunches of grapes. Staphylococcus aureus, famous for causing various skin infections, boils, and more serious systemic infections, is the classic example. Think staph, like a staff or bunch.
Grapes, chains, pears. Okay, so even within the spheres, there's a lot of diversity in how they stick together. What about the rod shapes, the bacilli?
Bacilli are, rod-shaped cells. They can vary quite a bit in length and width. Some may be very short and plump, almost resembling coccais. We call these coccobacilli. Like cocci, they can also be arranged in patterns. Some form chains, known as streptobacilli, where the rods remain attached end to end after division. Bacillus anthracis, the cause of anthrax, can form long chains. Bacilli can also show varied end shapes. Some are flat-ended, some rounded, some even cigar-shaped or bifurcated split. Some bacteria, like Corynebacterium diphtheriae, which causes diphtheria, have unique arrangements. Due to a sort of snapping division, they often lie at angles to each other, forming V and L shapes, sometimes called Chinese letter or cuneiform arrangements. It's quite distinctive. And importantly, some bacilli are spore formers, capable of producing those highly resistant endospores we mentioned. Genera like Bacillus and Clostridium are famous examples. Clostridium botulinum, which produces the toxin responsible for botulism, is a spore-forming bacillus.
Okay, rods and spheres. And then there are the more complex spiral forms. These sound like they might move differently too, based on their shape.
Precisely. Motility is often linked to these shapes. This category includes several types. Vibrios are curved or comma-shaped rods. They often have a single polar flagellum and exhibit a characteristic rapid, darting motility, sometimes described as vibratory, hence the name Vibrio. Vibrio cholerae, the cause of cholera, is the classic example. Then there are spirilla. Spirillin is singular spirilicins. These are rigid spiral or helical forms. They are typically longer than vibrios and have external flagella, usually at the ends, which they use for motility, rotating like corkscrews. Helicobacter pylori, which is associated with stomach ulcers and gastritis, is often described as a curved or spiral rod. Finally, we have the spiripedes. These are truly fascinating. They are long, slender, helical, and flexible spiral forms. Examples include Treponema pallidum, the pathogen behind syphilis, and Borrelia burgdorferi, which causes Lyme disease. What's unique about Spirochetes is their method of motility. They don't have external flagella. Instead, they possess flagellum-like structures called axial filaments or endoflagella, which run lengthwise between the bacterial inner membrane and outer membrane within the periplasmic space. The rotation of these filaments causes the entire cell to flex and rotate like a corkscrew, allowing them to move effectively even in viscous environments like mucus or tissues. They can literally bore through tissues.
Wow, that's a clever way to move. It sounds like some bacteria are also a bit shape-shifty, not always sticking to one perfect form.
They can be, yes. We call these pleomorphic bacteria. Pleomorphic just means having variable shapes. They might appear as a mixture of coccal and bacillary forms within the same culture, or show considerable variation in size and shape from cell to cell. This pleomorphism can occur naturally in some species, or it can be induced by factors like aging of the culture, nutrient limitations, or exposure to antagonistic substances like certain antibiotics that interfere with cell wall synthesis. And just to add another layer of diversity, there are the actinomycetes. These are true bacteria, but they grow in branching filamentous forms that superficially resemble fungi. They're important soil bacteria, and some produce antibiotics. Just shows the incredible morphological diversity within the bacterial world.
So if these outer layers and shapes are their interface with the world, what's powering the machine inside? Let's peel back that cell wall and dive into the bacterium's bustling internal world, the cytoplasm and its contents, where all the real work of life happens.
Absolutely. Let's go inside. The cytoplasm is essentially the substance filling the cell, enclosed by the cell membrane. It's mostly a viscous, watery solution, about 4/5 water, but it's packed with essential molecules. It contains enzymes needed for metabolism, nutrients being processed, waste products waiting to be expelled, dissolved gases, and crucially, the structures needed for growth and replication. Its vital role is to be the site where most of the important functions of the cell happen: growth, metabolism, replication. But importantly, and unlike eukaryotic cytoplasm, prokaryotic cytoplasm lacks those complex membrane-bound organelles like mitochondria, endoplasmic reticulum, Golgi apparatus, and lysosomes. It also doesn't exhibit cytoplasmic streaming, that directed flow of cytoplasm seen in larger eukaryotic cells. It's a simpler yet incredibly efficient internal environment.
So no fancy organelles floating around keeping things compartmentalized. But what about the essential machinery for making proteins? Every living cell needs that, right? That's got to be critical.
Absolutely critical, and that's where ribosomes come in. These are the protein synthesis factories essential in all living organisms. Ribosomes are actually the most numerous intracellular structures in most actively growing bacteria. A single E. coli cell can contain tens of thousands of them, and here again is that key difference we touched on earlier. All prokaryotes have the smaller 70S ribosomes, which are composed of a large 50S subunit and a small 30S subunit. Eukaryotes, in their cytosol, contain larger 80S ribosomes made of 60S and 40S subunits. This isn't just a minor structural detail, it's a fundamental difference that forms the very basis of how many of our most effective and life-saving antibiotics work. They selectively target and inhibit the bacterial 70S ribosome function, stopping protein production in the bacteria without significantly harming our own 80S ribosomes. It's a perfect example of selective toxicity.
It's amazing how a slight structural difference at that molecular level could be so strategically important for medicine. What else is floating around in the cytoplasm that keeps the cell going? Maybe like storage units.
Yeah, bacteria often store reserve materials in what are called cytoplasmic inclusions or nutrient granules. These are generally considered non-living components of the cell. They don't have metabolic activity themselves and are typically not bounded by membranes like organelles are. They essentially serve as internal storage depots. This allows bacteria to store excess nutrients when they're abundant for use during times of scarcity. Common examples include storing excess carbon in polymers like polyhydroxyalkanoates, PHAs, which are like bacterial plastics, or glycogen, similar to how animals store glucose. Other common inclusions can be lipid droplets, crystals of inorganic compounds like sulfur or phosphate, sometimes called velutin granules or metachromatic granules, and various pigments. Some aquatic bacteria also have gas vacuoles. These are remarkable structures, aggregates of gas-filled vesicles that help regulate buoyancy, allowing the bacteria to float or sink to optimal depths in water columns to access light or nutrients, like tiny biological submarines adjusting their ballast.
Okay, storage granules, gas vacuoles, and where does the actual blueprint for the bacterium reside? We said no nucleus, so where's all that crucial genetic information stored?
That's contained within the nucleoid, sometimes called the nuclear region or nuclear body. This is the irregularly shaped region within the cytoplasm where the main bacterial genetic material, the chromosome, is located. As we emphasized, unlike eukaryotes, this bacterial DNA is not enclosed inside of a membrane-bound nucleus. It's just concentrated in this specific area of the cytoplasm. Most bacterial DNA exists as a single, large, supercoiled, circular molecule of double-stranded DNA. This is referred to as the bacterial chromosome. A nucleoid is not considered a true organelle, and the DNA within it is typically not associated with histone proteins, the packaging proteins found in eukaryotic chromosomes, though bacteria have other nucleode-associated proteins that help organize the DNA. All of the cell's essential genetic information for structure, function, and reproduction is contained within this chromosome. And remember, bacteria are generally haploid, meaning they have just a single copy of this chromosome and therefore a single copy of each essential gene.
And often, you mentioned, they have these little extra bits of DNA too, these plasmids, that play a huge role in their adaptability and evolution, especially in response to us.
Yes, plasmids. These are really important players in the bacterial world. They are small, typically circular, double-stranded DNA molecules that exist separately from and replicate independently of the main bacterial chromosome. They are considered extrachromosomal DNA. While they are similar in structure to the main chromosome, circular double-stranded DNA, they are considerably smaller, usually carrying only a few genes to maybe a few dozen, compared to the thousands on the main chromosome. Crucially, plasmids are generally not essential for the bacterium's basic survival under normal conditions. They don't carry genes involved in core functions like basic metabolism or reproduction. Instead, they often carry genes that confer advantageous but non-essential traits to their bacterial host. The most well-known examples are genes for antibiotic resistance. Other examples include genes for virulence factors, like toxins or adhesion proteins that help cause disease, resistance to heavy metals, or the ability to metabolize unusual nutrient sources. Plasmids can be easily gained or lost by a bacterium. And importantly, they can be transferred relatively easily from one bacterium to another, even between different species, through processes like conjugation, mediated by sex pillar. This is a major form of horizontal gene transfer.
Horizontal gene transfer, meaning they can share genes sideways, not just vertically from parent to offspring.
Exactly. And this has huge implications for how quickly traits like antibiotic resistance can spread through bacterial populations. A resistance gene on a plasmid can rapidly disseminate, making infections much harder to treat. Plasmids are key drivers of bacterial evolution and adaptation.
This really raises an important question, doesn't it? How does the relative simplicity of a prokaryotic cell's internal organization compared to a complex eukaryotic cell actually work to their advantage? Why is that simplicity, combined with these flexible features like plasmids, actually a powerful adaptive strength for bacteria, allowing for such rapid evolution and diversification? It seems almost counterintuitive that less complexity could be better sometimes. Okay, now let's talk about the fortifications, the outer defenses that protect bacteria from their often harsh environments. Things like osmotic pressure changes, or even attacks from our immune system or antibiotics. This cell envelope is actually surprisingly complex in bacteria, isn't it? More so than in many other organisms.
That's a great way to put it, fortifications. The cell envelope is absolutely critical for bacterial survival, and it typically consists of the cell membrane, the cell wall, and sometimes an outer capsule or slime layer. Let's start from the inside out. First, we have the cell membrane, also known as the cytoplasmic membrane or plasma membrane. It's a phospholipid bilayer, very similar in basic structure to eukaryotic cell membranes, and it's located just inside the cell wall, or it's the outermost layer in bacteria that lack a cell wall, like mycoplasma. Its general functions are also similar to eukaryotic membranes. It acts as a selective permeability barrier. It carefully regulates the transport of molecules, nutrients coming in, waste products going out, using various transport proteins embedded within it. But prokaryotic membranes have a unique and vital additional function. They are also deeply involved in energy conservation. They serve as the location where a proton motive force is generated, a gradient of protons across the membrane. This gradient is essential for driving ATP synthesis, making cellular energy, powering flagellar rotation, and transporting certain molecules. Think of it like a tiny biological hydroelectric dam built into the membrane.
So they use their membrane for energy production, unlike our cells, which use mitochondria for that.
Precisely. Since they lack mitochondria, bacteria carry out cellular respiration and ATP synthesis using enzyme systems located in their cell membrane. It's a key difference. Another difference is that unlike eukaryotic membranes, which contain sterols, like cholesterol for stability, bacterial cytoplasmic membranes generally lack sterols. The exception is the mycoplasma group, which incorporates sterols from their environment into their membranes since they lack a cell wall.
Okay, so the membrane is the barrier and energy site. Then outside of that, for most bacteria, comes the star of the show, the cell wall. This isn't just a simple boundary, is it? It's fundamentally important and unique to bacteria.
The cell wall is absolutely fundamental. It's a rigid, multi-layered structure that lies immediately outside the cytoplasmic membrane in almost all bacteria. While it's porous and generally permeable to small molecules, its main roles are structural integrity and protection. It's responsible for maintaining the characteristic shape of the bacterium, whether it's coccus, bacillus, or spiral. And critically, it protects the cell from osmotic lysis. Bacteria often live in environments where the solute concentration outside the cell is lower than inside. Water tends to rush in, and without the rigid cell wall counteracting this pressure, the cell would swell and burst. And the unique and crucial component that gives the bacterial cell wall its strength and rigidity is peptidoglycan, also called murine.
Peptidoglycan. You mentioned this before. This is the stuff that's unique to bacteria, right?
Exactly. Peptidoglycan is absent in archaea and eukaryotes, making it an excellent target for antibiotics. It's responsible for the rigidity of the bacterial cell wall and for determining the cell shape. Its structure is quite remarkable. It's made up of a polysaccharide backbone consisting of alternating sugar derivatives. N-acetylmuramic acid, NAM, and N-acetylglucosamine, NAG, linked together in long chains. These parallel glycan chains are then cross-linked by short peptide bridges that extend from the NAM residues. What's particularly clever and biochemically unique is that these peptide crosslinks often contain unusual D-form amino acids, in contrast to the L-form amino acids found in virtually all proteins.
D-form amino acids. Why is that significant?
It's significant because most proteases enzymes that break down proteins, including those produced by host organisms as defenses, are specific for L amino acids. Using D amino acids makes the peptidoglycan structure much more resistant to degradation by host enzymes, contributing to its robustness. And this unique structure has profound significance in human health. Since the peptidoglycan cell wall is absolutely required for the survival of most bacteria but is completely absent in eukaryotic cells like ours, it represents an ideal target for selective toxicity. Several of our most common and effective antibiotics, notably the lactam antibiotics like penicillins and cephalosporins, work specifically by interfering with the enzymes that build these peptide crosslinks in peptidoglycan synthesis. They weaken the cell wall, leading to bacterial death, while having essentially no effect on human cells. It's a perfect example of exploiting a unique bacterial vulnerability.
That's brilliant. So this unique cell wall structure leads us directly to the Gram stain, doesn't it? That fundamental staining technique that quickly categorizes bacteria based on how their wall reacts.
Absolutely. The differential staining outcome of the Gram stain is directly related to the differences in cell wall structure, primarily the thickness of the peptidoglycan layer and the presence or absence of an outer membrane. This divides most medically important bacteria into two large groups, gram-positive and gram-negative. Gram-positive bacteria have remarkably thick cell walls, typically 15 to 80 nanometers thick. The peptidoglycan layer constitutes a significant portion, almost 95% of this thick wall. It's a dense mesh-like structure. Because of this thick, dense layer, when gram-positive cells are stained with the primary stain, crystal violet, and then treated with iodine, which acts as a mordant, forming a large complex with the dye, this complex gets trapped within the thick peptidoglycan layer. Even when decolorizing agent, like alcohol or acetone, is applied, the dye complex isn't easily washed out, so they retain the purple crystal violet stain and appear purple or blue under the microscope. Their cell walls also contain other molecules like teichoic acids and lipoketoic acids embedded within the peptidoglycan, which play roles in cell shape, division, and adhesion.
Okay, so thick peptidoglycan wall traps the purple stain. What about gram-negatives then? Why'd they end up pink?
Gram-negative bacteria have a much more complex cell envelope structure, but their peptidoglycan layer is comparatively very thin, maybe only 2 nanometers thick, located in the periplasmic space between the inner and outer membranes. Because this peptidoglycan layer is so thin, it doesn't effectively trap the crystal violet iodine complex. When the decolorizing agent is applied, it easily washes the purple dye complex out of the thin peptidoglycan layer. The cells become colorless at this stage. Then, when the counterstain, usually safranin, which is pink or red, is applied, the decolorized gram-negative cells pick up the second stain and appear pink or red.
And gram-negative bacteria, you mentioned, have this additional outer membrane outside the thin peptidoglycan layer. What does that extra layer bring to the table, both for the bacterium and for our understanding of how to fight them?
That outer membrane is a critical feature of gram-negative bacteria and makes them fundamentally different from gram-positives in many ways. It's a complex structure itself, an asymmetric bilayer. Its inner leaflet is composed mainly of phospholipids, similar to the cytoplasmic membrane, but its outer leaflet is primarily composed of lipopolysaccharide, LPS. This outer membrane acts as an additional protective barrier, making gram-negative bacteria generally more resistant to certain antibiotics, like penicillin G, which has trouble crossing it, and digestive enzymes, like lysozyme. However, it still needs to allow necessary nutrients in. So the outer membrane contains specialized proteins called porins, which form channels that allow for the passive diffusion of small hydrophilic molecules like ions, sugars, and amino acids across the outer membrane into the periplasmic space.
The periplasmic space. That's the gap between the two membranes. Does anything important happen there?
Yes, the periplasmic space is the region between the outer membrane and the cytoplasmic inner membrane in gram-negative bacteria. It's not just an empty gap. It's a gel-like compartment containing the thin peptidoglycan layer and a variety of proteins and enzymes. These include enzymes involved in nutrient acquisition and transport, proteins involved in chemotaxis, sensing the environment, and importantly, enzymes like violactamases. These vitamases can degrade vitamic like penicillin before they even reach their target, the enzymes involved in peptidoglycan synthesis near the inner membrane. This is a major mechanism of antibiotic resistance in gram-negative bacteria.
Okay, and you mentioned LPS as a major component of that outer membrane in gram-negatives. What's its significance for us when we get infected? You said it was toxic.
Lipopolysaccharide, or LPS, is an integral component of the outer leaflet of the outer membrane of gram-negative bacteria, and yes, it's a very big deal biologically and medically. LPS is extremely potent biologically and is commonly called endotoxin. Endo, because it's part of the bacterial cell structure, typically released in significant amounts only when the bacteria lies or die. It's also called a pyrogen because it directly induces fever. LPS is responsible for many of the severe symptoms associated with infections by gram-negative bacteria, such as fever, chills, muscle aches, a drop in blood pressure, and in severe cases, disseminated intravascular coagulation, DIC, and life-threatening septic shock. Our immune system mounts a very strong, sometimes dangerously excessive inflammatory response to LPS. It's important to remember that gram-positive bacteria do not produce endotoxins because they lack LPS and the outer membrane structure. They may produce exotoxins, secreted proteins, but not endotoxin. LPS itself is a complex molecule consisting of three distinct parts. Lipid A, which is the fatty acid-containing portion embedded in the membrane and is responsible for virtually all of its potent endotoxic activity. A core oligosaccharide, a short chain of sugars linking lipid A to the third part, and the O-polysaccharide, also known as O-antigen, which is a long repeating chain of sugars extending outwards from the bacterial surface. The composition of the O-antigen is highly variable between different strains and species of gram-negative bacteria and contributes to their antigenic properties. It's what our immune system often recognizes, or fails to recognize if it's disguised.
Wow, so that outer membrane is a barrier, contains channels, holds resistance enzymes, and has this potent toxin built right in. That's complex. Are there any other unique cell wall types we should know about that defy these gram-positive, gram-negative categories?
Yes, there are a few important exceptions or variations. The most significant group are the acid-fast bacteria, primarily the genus Mycobacterium, which includes the causative agents of tuberculosis and leprosy, and nocardia. Their cell walls are structurally distinct. They do have a peptidoglycan layer, but outside of that, they possess a thick, complex, waxy outer layer rich in unique lipids called mycolic acids. These mycolic acids form a very hydrophobic, impermeable barrier. This waxy layer makes them incredibly tough and resistant to many common disinfectants and antibiotics. It also makes them resistant to gram staining. They typically stain poorly or variably. They require a special staining procedure called the Ziehl-Neelsen stain or Kenyon stain, acid-fast staining, which uses heat or detergents to drive a primary stain, like carbofuscin, into the waxy wall. Once stained, they resist decolorization by strong acids, like acid alcohol, while other bacteria are decolorized, hence the name acid-fast. This unique wall structure is a major factor in the difficulty of treating diseases like TB.
Okay, so mycobacteria have that waxy wall. What about bacteria that might lose their wall or naturally lack one?
Right. Bacteria can sometimes exist in cell wall deficient CWD forms. This can happen if their peptidoglycan synthesis is interfered with by agents like the enzyme lysozyme, found in our tears and saliva, it breaks down peptidoglycan, or by certain antibiotics like penicillin.
Oh.
If the bacterial cell wall is entirely removed, for example from a gram-positive cell, the resulting wall-less cell is called a protoplast. If the cell wall is only partially removed, or if an outer membrane remains, as in gram-negatives, it's called a spheroplast. These forms are typically osmotically sensitive and spherical. Spheroplast formation can sometimes be reversible if the antibiotic is removed. The term L forms is also used, often referring to CWD organisms that can replicate, sometimes arising spontaneously or induced by antibiotics. And then there's the genus Mycoplasma. These bacteria are unique in that they are naturally occurring bacteria that completely lack a rigid cell wall. They don't have peptidoglycan at all. As a result, they have no definite shape. They are pleomorphic and are completely resistant to antibiotics that target cell wall synthesis. To compensate for the lack of a wall, their cell membrane is stabilized by incorporating sterols acquired from their host or environment.
Fascinating. And finally, for the outer layers, some bacteria have this extra coating, this capsule or slime layer, that can really determine how they interact with their host or environment. What's that about?
Yes, the capsule or slime layer, often collectively referred to as the glycocalyx, sugar coat, is an extracellular, sticky, gelatinous polymer secreted by many bacteria covering the surface of the cell, located outside the cell wall if present. It's usually composed of polysaccharides, long chains of sugars, although some bacterial capsules are made of protein, like the poly-D-glutamic acid capsule of Bacillus anthracis. Its functions are really critical, particularly in disease. It often acts as a major virulence factor and provides protection for the cell wall and the bacterium itself. One key role is protection from phagocytosis. The capsule often makes the bacterial surface slippery and masks surface components that immune cells, like macrophages, would normally recognize and bind to. This helps the bacterium evade engulfment and destruction by our immune system. Think of it like a bacterial cloaking device or shield. It also mediates adhesion. The sticky nature of the glycocalyx allows bacteria to attach firmly to other cells, like host tissues, or to inanimate surfaces like catheters, medical implants, or teeth. This attachment is often the crucial first step in colonization and infection, and it's essential for the formation of biofilms.
Biofilms. Those slimy communities of bacteria.
Exactly. Biofilms are structured communities of bacteria embedded in a self-produced matrix, largely composed of glycocalyx material. They are notoriously difficult to treat with antibiotics and are a major issue in persistent infections, like on medical devices or in chronic wounds. The glycocalyx forms the scaffolding of the biofilm. A slime layer can also sometimes be used as a reserve food source for the cell. Capsules can also be important for identification-specific antibodies against capsule polysaccharides. Serotyping can help identify strains of bacteria like Streptococcus pneumonia or Haemophilus influenza.
Is there a difference between a capsule and a slime layer?
Yes. It's mainly a matter of organization and attachment. A condensed, well-defined, firmly attached layer is generally called a capsule. It's relatively impermeable and typically excludes particles like India ink, making it visible as a clear halo in negative staining. A loose, diffuse, easily deformed, and loosely attached layer is usually called a slime layer. Examples of important capsule-forming bacteria include Streptococcus pneumonia, capsule is essential for virulence, Klebsiella pneumonia, Haemophilus influenza type B, Hybe vaccine targets the capsule, Neisseria meningitis, and Bacillus anthracis. These capsules can be demonstrated through various methods, including gram staining, where they may appear as a clear zone, India ink staining, negative staining, specialized capsule stains, electron microscopy, and serological methods using specific antibodies.
Okay, so if we connect all this back to the bigger picture, you can really see how the bacterial cell wall is literally a bacterial Achilles heel, right? Targeted by so many of our life-saving antibiotics because it's unique to them. And the capsule, on the other hand, is like their shield or cloaking device, helping them evade our immune systems and stick to surfaces, leading to those persistent infections and hard-to-eradicate biofilms. These outer layers are critical battlegrounds. Beyond just protection and sticking, bacteria have evolved these incredible appendages that allow them to navigate their microscopic worlds, either by moving purposefully towards something good or away from something bad, or by attaching firmly to specific surfaces. These are really sophisticated biological machines on a tiny scale.
Absolutely. Let's start with the structures responsible for movement. Flagellus, singular, flagellum. These are long, thin, whip-like filaments that extend out from the bacterial cell surface. They are primarily composed of a protein called flagellin, arranged in a hollow helical structure. Their main function is mediating bacterial motility, allowing bacteria to swim through liquid environments. They act like tiny propellers, rotating to push or pull the bacterium along. This motility is often directed. Bacteria can move towards attractants, like nutrients, or away from repellents, like toxic chemicals, in a process called chemotaxis. Structurally, a bacterial flagellum is quite complex, originating from the bacterial protoplasm, cytoplasm, and membrane, and extending outwards. It has three main parts. First, the filament. This is the long helical screw-like part that extends into the environment and actually does the propelling. Second, the hook. This is a short, curved, flexible structure that connects the filament to the basal body. It acts like a universal joint, allowing the filament to rotate freely and point away from the cell body. And third, the basal body. This is the intricate motor assembly that anchors the flagellum into the cell envelope, cell wall, and membranes, and drives its rotation. It consists of a central rod surrounded by a series of protein rings that act like bearings.
A motor assembly. So it's literally a tiny biological rotary motor. And you mentioned earlier that gram-positive and gram-negative bacteria even differ in their flagellar structure down there at the base.
They do, reflecting their different cell envelope structures. Gram-positive organisms, with their thick peptidoglycan glycan wall but only one membrane, have a simpler basal body with typically two rings, one embedded in the peptidoglycan layer and one anchored in the plasma membrane. Gram-negative organisms, with their more complex envelope, inner membrane, thin peptidoglycan outer membrane, have a more elaborate basal body with four rings. The L ring associated with the lipopolysaccharide outer membrane, the P ring associated with the peptidoglycan layer, and the M and S rings embedded in or associated with the inner plasma membrane. It's a more complex anchoring system for the more complex wall. And what's truly remarkable is how this motor is powered. The bacterial flagellum is driven by a rotary engine formed by proteins like the MOT complex located at the anchor point in the cell membrane. This engine is powered directly by the proton motive force we discussed earlier, the flow of protons, hydrogen ions, across the bacterial cell membrane, down the concentration gradient established by the cell's metabolism. It's an astonishing feat of molecular engineering, converting electrochemical potential energy directly into mechanical rotational energy, causing the filament to spin at incredible speeds, sometimes over 100,000 RPM.
Wow, powered by proton flow. That's incredible.
Right.
And bacteria arrange these flagella in different ways on their surface, right?
Oh.
Which presumably affects how they move.
Yes, the number and arrangement of flagella, known as the flagellar arrangement scheme, are characteristic features used in classifying bacteria. There are several main patterns. Some are monotrichous, meaning they have a single flagellum, typically located at one end pole of the cell, like Vibrio cholerae. Others are amphitricious, having either a single flagellum or a tuft of flagella at both ends of the cell, like Alcaligenes faecalis. Lophotricious bacteria have a tuft or bundle of flagella arising from one end of the cell, seen in some spirilla. And paratricious bacteria have flagella distributed more or less randomly all over the entire surface of the bacterial cell. E. coli and Salmonella are common examples. These bacteria typically move in a run-and-tumble fashion, coordinating their flagella rotation. It's also worth noting that most Kochi, the spherical bacteria, are non-motile and lack flagella altogether. We can observe bacterial motility and infer the presence of flagella using techniques like the hanging drop method, watching live bacteria swim under a microscope, observing swarming growth on semi-solid agar media, or specialized methods like Craigy's tube method.
Okay, so flagella are for swimming. What about those other, often shorter hair-like structures we sometimes see covering the surface of bacteria? The polyli or fimbriae, what's their function if not movement?
Right, those are fimbriae, or pili, singular pilosus. These are also proteinaceous appendages extending out from the cell surface, but they are generally distinct from flagella. They are typically shorter, thinner, straighter, and much more numerous, sometimes covering the entire cell surface like bristles. They are made of a protein called pleelin. Their primary function is generally not locomotion, although there are a few exceptions, like type IV pili, used for a kind of twitching or gliding motility in some bacteria like Mysococcus or Pseudomonas. Instead, fimbriae and pili are mainly involved in adhesion. They act like tiny grappling hooks or Velcro, facilitating the crucial attachment of a bacterium to surfaces. This could be attachment to other bacterial cells, to inanimate surfaces like rocks, soil particles, or medical devices, initiating biofilm formation, or importantly, to host cells, like the epithelial cells lining our respiratory or intestinal tracts during pathogenesis. These adhesins are often essential for the virulence of many bacterial pathogens, allowing them to colonize host tissues and resist being washed away by fluid flow, like urine or mucus. If they can't stick, they often can't cause disease.
So they're primarily for sticking. Are there different types of pili?
Yes, we generally distinguish between two main types. Common pili or fimbriae are primarily involved in adhesion to surfaces or host cells. Then there are specialized sex pili, also called F pili or conjugation pili. These are typically longer and less numerous than common fimbria. Their specific function is to initiate contact between two bacterial cells during the process of bacterial conjugation, forming a bridge that allows the transfer of genetic material, usually plasmid DNA, from a donor bacterium to a recipient bacterium. Sex pilis are essential for this important mechanism of horizontal gene transfer.
It's truly astonishing how sophisticated these tiny biological machines are, operating at the nanoscale. Imagine the precision required for a bacterial flagellum to rotate like a propeller powered by proton flow, or the strategic advantage conferred by pilacy for sticking firmly to just the right surface, which is such a key step in establishing many bacterial infections. This really raises an important question. How do these specific structures relate to the different types of infections bacteria cause, and how might targeting these structures, like blocking adhesion or inhibiting motility, be a viable strategy for developing new anti-infective treatments beyond traditional antibiotics? Now, prepare to be amazed, because we're going to talk about one of bacteria's most incredible adaptations, something that allows them to withstand conditions that would annihilate almost any other form of life. These are like bacterial survival pods, truly the ultimate biological time capsules.
You must be talking about bacterial endospores. And yeah, time capsules is a great way to describe them. These tiny structures are marvels of biological engineering, literally designed to survive extreme adversity, almost like a biological apocalypse. How do they do it? It really is all in their incredible, complex, multi-layered design. So what is an endospore? It's a dormant, tough, and non-reproductive structure produced internally, hence endo, by certain types of bacteria, primarily from the gram-positive genera Bacillus, which are typically aerobic or facultative, and Clostridium, typically obligate anaerobes. The key thing is that they are metabolically inert. They have essentially zero metabolic activity. They're not growing, not respiring, not doing much of anything except waiting. Endospore formation, a complex developmental process called sporulation, is usually triggered by environmental stress, most commonly a lack of essential nutrients like carbon or nitrogen depletion, or other adverse conditions like extreme temperatures or pH. It's a survival strategy, not a reproductive one.
Not reproductive, so one bacteria makes one spore.
Exactly. During sporulation, the bacterium essentially divides within its own cell wall, packaging a copy of its chromosome and essential cellular components into a highly protected core, which then matures into the spore. The original vegetative cell, the mother cell, eventually disintegrates, releasing the mature endospore. Later, under favorable conditions, that single spore can germinate and give rise to a single vegetative bacterium again. So it's one cell in, one spore out, one spore germinates back into one cell. It's about survival through time, not increasing numbers.
So they're just waiting out the storm, effectively, for potentially incredibly long periods, centuries even.
That's right. And they are astonishingly good at it. Their resistance is extreme, far exceeding that of the vegetative cells they come from. Bacterial spores are highly resistant to a vast array of environmental stresses that would quickly kill most other life forms. This includes things like intense heat. Many can survive boiling water for extended periods, harsh UV radiation, gamma radiation, chemical disinfectants like alcohols, phenols, even hydrogen peroxide to some extent, extreme desiccation, drying, pressure, and various biocides. They can remain dormant and viable for extraordinarily long periods, years, decades, centuries, potentially even longer under the right conditions, just waiting for favorable conditions to return.
How on earth do they achieve such incredible resilience with such a tiny structure? What's the secret to their toughness?
It all comes down to their complex, multi-layered structure and unique chemical composition. Think of it like a series of nested, highly specialized protective shells around the precious cargo, the DNA and core machinery. Starting from the outside, there might be a thin outer covering called the exosporium, which can play roles in adherence and maybe some biocide protection. Inside that is the spore coat, a thick layer made primarily of highly cross-linked keratin-like proteins. This coat acts like a molecular sieve, excluding large toxic molecules like lysozyme and providing resistance to many toxic chemicals and enzymes. It's a major protective barrier. It may also contain enzymes needed later for germination. Beneath the spore coat is the cortex. This is a very thick layer consisting mainly of a unique type of loosely cross-linked peptidoglycan. The cortex plays a crucial role in maintaining the dehydrated state of the core and contributes significantly to the spore's resistance to heat and biocides. Inside the cortex is the core wall, which surrounds the central protoplast, or core, of the endospore. This core wall contains normal peptidoglycan that will become the cell wall of the new vegetative cell upon germination. And finally, the core itself. This contains the bacterial DNA, chromosome, ribosomes, and essential enzymes, but it's in a highly dehydrated state. Much of the water has been removed. It also has a very high concentration of a unique chemical called calcium dipicolinate, KDPA, which complexes with calcium ions and makes up a significant portion of the spore's dry weight. This KDPA complex is thought to intercalate into the DNA, stabilizing it against heat denaturation, and the overall low water content makes proteins and DNA much more resistant to heat, radiation, and chemical damage.
Wow, multiple layers, unique chemicals, dehydration. It's like a microscopic fortress. How do we even see these things if they're so tough and resistant to regular stains?
That's a good point. Visualizing endospores with standard light microscopy using common stains, like the Gram stain, can be difficult. Because their spore coat is so impermeable, stains don't easily penetrate. The vegetative part of a bacterial cell might stain, for example, purple if gram-positive, but the endospore within it, or a free spore, is often left unstained and appears as a clear, refractive oval or sphere within the colored cell or against the background. To specifically visualize endospores, special staining techniques are needed that use heat or strong chemicals to drive a primary stain, like malachite green, into the spore coat. Once stained, the spore resists decolorization, for example, by water, while the vegetative cells are decolorized and then counterstained with a different color, like safranin, making them pink red. The Schaefer-Fulton stain is a common method, resulting in green spores within pink vegetative cells. A molar stain is another method. The position and sometimes the shape of the endospore within the original bacterial cell before it's released can also be characteristic and useful for identification.
Position, so where the spore forms in the cell matters.
Yes. Spores can be central, located roughly in the middle of the cell, as seen in Bacillus cereus or Clostridium bifurmentans. They can be subterminal, located near one end, but not right at the tip, like in Clostridium perfringens or Bacillus subtilis. Or they can be terminal, located right at one end pole of the cell, as famously seen in Clostridium tetani, the pathogen that causes tetanus. In C. tetani, the terminal spore is often spherical and wider than the vegetative cell, causing the cell to bulge at that end, giving it a distinctive drumstick or tennis racket appearance. Spores themselves can also be oval or spherical in shape.
Okay, so they're incredibly resistant. If boiling doesn't reliably kill them, how do we actually destroy them, especially in critical settings like medicine or food safety, where sterility is paramount?
While they are significantly resistant to many stresses, they are not indestructible. Endospores can, of course, be destroyed by incineration, burning. More practically, in laboratories, healthcare settings, and the food industry, the most reliable method for destroying endospores and achieving sterilization is autoclaving. Autoclaving uses saturated steam under high pressure. This allows the temperature of the steam to exceed the boiling point of water, 100 degrees C. Standard autoclaving conditions are typically 121 degrees C, achieved at about 15 psi or 1 atmosphere above ambient pressure, maintained for a sufficient time, usually 15-20 minutes or longer depending on the load. The combination of high temperature and penetrating moist heat effectively denatures the proteins and damages the DNA even within the resistant spore core. Autoclaving is the gold standard for sterilizing surgical instruments, laboratory media, and other critical items. There's also an older, less commonly used indirect method called tyndallization, named after John Tyndall. This was historically used for sterilizing heat-sensitive liquids that couldn't withstand autoclaving temperatures. It involves cycles of heating, EVGM to 100 degrees C for a short time to kill vegetative cells, followed by incubation periods at room temperature to allow any surviving spores to germinate into more heat-sensitive vegetative cells, and then repeating the heating cycle several times over consecutive days. It essentially tricks the spores into germinating and then kills the resulting vegetative cells. It's much less reliable and more time-consuming than autoclaving.
And these spores even have a practical application for us, don't they? As a way to check if our sterilization process has actually worked.
Absolutely. Because endospores are the most resistant known forms of life, certain types of bacterial endospores, particularly those from highly heat-resistant thermophilic bacteria like Geobacillus stereothermophilus, formerly Bacillus stereothermophilus, are used commercially as biological indicators, BIs. These BIs contain a known number of viable spores on a carrier, like a paper strip, inside a vial, often with a growth medium and a pH indicator. They are placed inside the autoclave load during a sterilization cycle. Afterwards, the vial is incubated. If the sterilization process was successful and killed all the spores, the medium remains unchanged. If any spores survived, they will germinate and grow, metabolizing the medium and causing a color change due to pH shift. This provides direct biological proof that sterilizing conditions were achieved. It's a crucial quality control step in healthcare and industry to ensure patient safety and product sterility. And of course, reactivation of the endospore back into a growing cell occurs when conditions become more favorable again, presence of water, nutrients, suitable temperature. This process involves several steps. Activation, which may require a trigger, sometimes sublethal heat, germination, rapid loss of resistance properties, water uptake, swelling, shedding of spore coats, and outgrowth, synthesis of new components and development into a fully metabolically active vegetative cell ready to resume growth and division.
So, knowing all this about endospores, their incredible resistance, their longevity, how hard they are to kill, how does that fundamentally change how we think about things like food preservation, like canning, which needs high heat to kill Clostridium botulinum spores, or sterilization protocols in a hospital? It really makes you realize just how robust and persistent these tiny organisms can be and why stringent methods are so necessary. They are true survivors. Right, now that we've delved deep into what bacteria are, their history, their structure, their survival pods, let's explore how these incredible organisms actually multiply. It's a surprisingly simple process, yet it's incredibly effective and accounts for their ability to rapidly colonize environments or cause infections.
Indeed. The primary mode of bacterial reproduction is remarkably straightforward. It's called binary fission. This is a form of asexual reproduction, meaning it involves only one parent cell and doesn't involve the mixing of genetic material like sexual reproduction does. In binary fission, a single parent bacterial cell essentially grows larger, replicates its single circular chromosome, and then the cell divides into two genetically identical daughter cells. The process involves the chromosome attaching to the cell membrane, replication initiating, the cell elongating, the two chromosomes separating, and then a new cell wall and membrane forming down the middle, septum formation, to split the cell in two. This process results in a remarkably rapid, logarithmic or exponential growth rate under favorable conditions. One cell becomes two, two become four, four become eight, and so on.
Logarithmic growth. That sounds fast. And we often talk about generation time for bacteria. What does that actually mean?
Right. The generation time, also known sometimes as the doubling time, is defined as the specific time required for a single bacterium to divide into two daughter cells, or equivalently, for the entire bacterial population to double in number under optimal environmental conditions, like the right temperature, nutrients, pH, etc.
And this doubling time can vary wildly between different bacterial species, right? Which helps explain why some infections can seem to take hold almost overnight, while others develop much more slowly.
Absolutely, there's huge variation. To illustrate, Escherichia coli, a very common gut bacterium often used in labs, has an incredibly short generation time. Under optimal conditions, it can double in as little as 20 minutes. Think about that. In just 1 hour, one E. coli cell can become 8. In 2 hours, 64. In just a few hours, it can explode into millions or billions of cells. In stark contrast, Mycobacterium tuberculosis, the pathogen that causes TB, grows much more slowly. Its generation time is typically around 12 to 24 hours. This is one reason why TB develops slowly and requires long treatment courses. And Mycobacterium leprae, the bacterium that causes leprosy, is even slower. Its generation time is estimated to be as long as 14 to 20 days. This is why leprosy has such a long incubation period and is so slow to progress.
Wow, from 20 minutes to 20 days. There's an enormous range. But that incredibly rapid doubling time for many common bacteria really explains how a single contaminating bacterium, or just a few getting into a wound, can quickly explode into a massive population. It impacts human health dramatically, like in a rapidly progressing infection, but also industrial processes like using bacteria for fermentation in a remarkably short time frame. It's clearly a key to their immense success and adaptability on this planet. So if bacteria can divide so quickly, doubling every 20 minutes in some cases, why don't they just completely take over the entire world in a matter of hours or days? Something must limit them, right? The answer, I gather, lies in understanding their life cycle when grown in a contained environment, which is often represented by what's called a bacterial growth curve.
That's an excellent point, and yes, their growth isn't unlimited in the real world or even in the lab usually. Bacterial growth is most often studied in what's called a batch culture. This is the most common laboratory growth method. In this setup, you inoculate a small number of bacteria into a closed vessel like a flask or test tube containing a single, finite batch of sterile liquid growth medium. You then incubate it under suitable conditions. As the bacteria grow and divide by binary fission, they consume the nutrients in the medium and also excrete waste products into it. Eventually, nutrients become scarce, and toxic waste products accumulate, which inevitably limits further growth and leads to changes in the population dynamics over time. When we plot the number of viable living bacteria in the population over time in such a batch culture, we typically see a characteristic pattern with 4 distinct phases. This plot is known as the bacterial growth curve, and it gives us a kind of roadmap of a bacterial population's life in a closed system.
Okay, 4 phases. Tell us about the first phase. What happens when the bacteria are just getting started in their new home, the fresh medium?
That's the lag phase. This is the initial period right after inoculation. During this phase, the bacteria are adapting themselves to the new growth conditions. They might need to synthesize new enzymes to utilize the available nutrients or repair any damage incurred during transfer. So it's not typically a period of active cell division. The population size doesn't increase much or at all, but it is a period of intense metabolic activity. Individual bacteria are growing in size, maturing, synthesizing essential components like RNA, enzymes, and various metabolites needed to prepare for rapid growth. The cells are getting ready to divide. The duration of this lag phase can vary quite a bit, from just minutes to several hours, depending on the bacterial species, the age and condition of the inoculum culture, and how different the new medium is from the previous one. Think of it as their necessary warm-up or adjustment period before the race begins.
Okay, so they're adapting, getting ready,
Yeah.
and then they kick into high gear. What's the next phase?
Precisely. After the lag phase, they enter the log phase, which is short for logarithmic phase, also sometimes called the exponential phase. This phase is characterized by rapid and consistent cell division by binary fission. During the log phase, the number of new bacteria appearing per unit time is proportional to the present population size. Growth is balanced. All cellular components are being synthesized at constant rates relative to each other. The population doubles at regular intervals, dictated by the generation time for that specific bacterium under those specific conditions. If you plot the logarithm of the cell number versus time, this phase appears as a straight line with a positive slope. This is the period of most rapid growth. During this phase, the cells are typically at their healthiest, smallest, and most uniform in terms of size and staining properties. They are also generally most susceptible to antibiotics that target active processes like cell wall synthesis or protein synthesis. However, as you pointed out, this exponential growth cannot continue indefinitely in a closed batch culture. Why? Because the growth medium is soon depleted of essential nutrients, and or inhibitory waste products, like organic acids from fermentation, begin to accumulate to toxic levels.
Right. Resources run out, waste builds up. Which brings us to the next phase, where things start to level out, suggesting some kind of limitation is really kicking in.
Exactly, that's the stationary phase. During this phase, the overall bacterial growth rate slows down significantly, and eventually, the rate of new cell production essentially balances the rate of cell death. The net result is that the total number of viable bacteria in the population remains relatively constant, or reaches a plateau. This plateau is often reached due to a growth-limiting factor, such as the depletion of an essential nutrient, the accumulation of inhibitory waste products to a critical level, or maybe lack of oxygen if it's an aerobe in a poorly aerated culture. The population has reached the maximum density, the carrying capacity that the environment can support. On the growth curve graph, log of cell number versus time, this phase appears as a smooth horizontal line. Cells in the stationary phase often undergo physiological changes to adapt to survival under stress. They might become smaller, their metabolism slows down, they express stress response genes. And importantly, for spore-forming bacteria like Bacillus or Clostridium, endospore formation sporulation often begins during the late log phase or early stationary phase as a survival mechanism triggered by nutrient depletion or other unfavorable conditions. It's their way of creating a lifeboat for the population when conditions get tough.
Okay, so they hit a plateau where growth equals death. But eventually, conditions must get even worse in that closed flask, right? What happens then? Sadly for the bacteria, I suppose, comes the decline.
Yes, inevitably, in a closed batch culture, the stationary phase is followed by the death phase, sometimes called the decline phase. In this final phase, the rate of cell death exceeds the rate of any residual cell division, and the number of viable bacteria in the population begins to decline, often exponentially, though sometimes more slowly or irregularly. This decline is primarily due to the continued lack of essential nutrients, the accumulation of toxic metabolic byproducts to lethal levels, or increasingly unfavorable living conditions like drastic pH changes or oxygen depletion. The environment simply becomes too hostile for survival. Bacteria essentially die faster than they can multiply, leading to a decrease in the viable cell count over time.
So this classic four-phase growth curve lag, log, stationary death describes what happens in a closed system, a batch culture. Does it differ significantly in other environments, like within our bodies during an infection or in industrial fermenters?
It can differ, yes. While batch culture is the most common laboratory method and gives us this fundamental model, other systems exist. For example, in industry or research, sometimes continuous culture systems are used, like a chemostat or turbidostat. In these systems, fresh medium is continuously added to the culture vessel at a steady rate, while an equal volume of spent medium in cells is removed. This allows bacteria to be maintained in a state of prolonged exponential log phase growth at a controlled rate and cell density by constantly replenishing nutrients and removing waste. This is very useful for industrial production of things like enzymes or antibiotics, or for research requiring cells in a specific physiological state. Within our bodies during an infection, the situation is much more complex and dynamic than a simple batch culture. Bacterial growth might exhibit elements of all these phases in different locations or at different times. Nutrient availability fluctuates, waste products might be removed by blood flow, and crucially, the host's immune system is actively trying to eliminate the bacteria. So, the growth curve in vivo is often much more irregular and influenced by this constant host-pathogen interaction, but understanding the basic principles of the bacterial growth curve from batch culture is still incredibly critical. It helps us understand microbial physiology, optimize industrial processes, and importantly, in medicine, it informs our strategies for treating infections, for example, knowing that antibiotics targeting growth are often most effective during the log phase, or understanding how bacteria might survive in a dormant state during the stationary phase.
That makes sense. Like all living organisms, bacteria require a precise set of nutrients to fuel all that metabolic activity and drive their incredibly rapid cell division, especially during that log phase. So what are their essential ingredients for life? What do they need to eat, essentially? And how do those needs dictate where they can live and thrive?
Right, nutrition is fundamental. Bacteria, despite their diversity, share some basic nutritional requirements. We can think about these in terms of essential elements and the forms they need them in. First, the essential nutrients, the absolute must-haves. These include water, absolutely essential, making up the bulk of the cell and serving as the solvent for all biochemical reactions. Urban source. Carbon is the backbone of all organic molecules. Bacteria vary greatly here. Autotrophs, like cyanobacteria or some soil bacteria, can use inorganic carbon dioxide, CO2, from the atmosphere as their sole carbon source, fixing it into organic compounds, often using light energy, photoautotrophs, or chemical energy, chemoautotrophs. Heterotrophs, which include most bacteria and certainly all human pathogens, require preformed organic compounds from their environment as their carbon source. They use things like sugars, glucose, amino acids, lipids, etc., both for building their own cellular components, biosynthesis, and often for energy production as well. Nitrogen source. Nitrogen is crucial for building proteins and amino acids and nucleic acids, DNA and RNA. Bacteria might get nitrogen from inorganic sources like ammonia, NH3, or nitrate, NO3, or from organic sources like amino acids or proteins in their environment. Some remarkable bacteria can even fix atmospheric nitrogen gas, N2, converting it into ammonia, a process vital for global nitrogen cycling. Inorganic salts. A variety of mineral ions are necessary, often in smaller amounts but still essential. These include phosphorus as phosphate for nucleic acids, ATP, phospholipids, sulfur for certain amino acids like cysteine and methionine and some vitamins, potassium, magnesium, calcium, iron, critical for cytochromes and enzymes, and various trace elements like manganese, zinc, copper, cobalt, molybdenum needed as cofactors for enzyme activity.
Okay, those are the basic elements. What about specific types of organic nutrients that many bacteria, especially the heterotrophs like pathogens, need?
Right. Beyond the basic elements, many heterotrophic bacteria need specific organic nutrients supplied. Carbohydrates, especially simple sugars like glucose, often serve as the primary and preferred energy source for many bacteria, broken down through glycolysis and respiration or fermentation. Amino acids are the building blocks for proteins. Some bacteria are quite self-sufficient and can synthesize all 20 standard amino acids from simpler precursors if given a basic carbon and nitrogen source. Others, however, are more fastidious. They lack some of the necessary biosynthetic pathways and must obtain certain essential amino acids preformed from their environment or host. Similarly, some bacteria require specific vitamins, purines, and pyrimidines. Vitamins often function as essential coenzymes needed for various metabolic reactions. Purines and pyrimidines are the building blocks for nucleic acids, DNA. Again, some bacteria can make their own, while others need them supplied. These required organic compounds that the bacterium cannot synthesize itself are often called growth factors.
So their dietary needs can be quite specific.
Exactly. This diversity in nutritional requirements helps explain why different bacteria thrive in such incredibly diverse environments. From the nutrient-rich human gut, exploiting our digested food, to the nutrient-poor open ocean, from hot springs relying on sulfur compounds to the soil breaking down complex organic matter. Their specific dietary needs and metabolic capabilities dictate where they can grow, what roles they play in different ecosystems like decomposition or nutrient cycling, and how they might cause disease by utilizing host resources. Understanding these needs is also crucial for culturing them in the laboratory we have to provide the right menu in the growth medium.
Beyond just food or nutrients, bacteria are also incredibly sensitive to their physical environment. You mentioned earlier they are master adapters, but they often have very specific preferences for things like temperature, oxygen levels, pH, and more. How do these physical factors regulate their growth, and what does that mean for us in terms of controlling them or understanding where they live?
These physical environmental factors are absolutely crucial regulators of bacterial growth and survival. They define the niche where a particular bacterium can thrive. Let's start with temperature. This is a really big one. Each bacterial species has a characteristic temperature range within which it can grow. There's a minimum temperature below which growth won't occur, an optimal temperature at which growth is most rapid, and a maximum temperature above which growth stops and the cell may be damaged or killed, often due to enzyme denaturation. Pathogenic bacteria, those that cause disease in humans and other warm-blooded animals, typically have their optimal growth temperature around human body temperature, which is about 37 degrees C, 98.6 degrees F. This makes sense. They're adapted to thrive inside us. Based on their preferred temperature ranges, bacteria are broadly grouped. Psychrophiles are the cold-loving bacteria. They grow best at relatively low temperatures, typically with optima below 15-20 degrees C, and some can even grow slowly near freezing point. They're found in cold environments like refrigerators, deep oceans, or polar regions. Some can cause food spoilage even under refrigeration. Mesophiles are the moderate-loving bacteria. This is the largest group and includes most bacteria we commonly encounter, including almost all human pathogens and the normal flora living on our bodies. They grow optimally in the moderate temperature range, typically between 20-45 degrees C, with optima often around 30-37 degrees C. Thermophiles are the heat-loving bacteria. They thrive at high temperatures with optima typically above 45 degrees C or 50 degrees C. Some extreme thermophiles or hyperthermophiles, often found in hot springs, volcanic vents, or compost heaps, can grow at temperatures near or even above boiling point. Their enzymes and membranes are specially adapted to withstand heat.
Okay, temperature is key. What about oxygen? That seems like another fundamental requirement. But it's not straightforward for bacteria, is it? Some need it, some hate it.
Exactly. Oxygen requirements are another critical classifying feature reflecting different metabolic strategies. Bacteria show a wide spectrum of relationships with oxygen. Obligate, or strict aerobes. These bacteria absolutely require oxygen for growth. They use oxygen as the final electron acceptor in aerobic respiration to generate energy efficiently. They cannot grow in the absence of oxygen. Examples include Pseudomonas aeruginosa, often found in lungs or burns, and Mycobacterium tuberculosis. Facultative anaerobes. These are highly versatile and adaptable. They can grow either in the presence or the complete absence of oxygen. If oxygen is available, they typically prefer to use it for aerobic respiration. It yields more energy. But if oxygen is absent, they can switch to alternative metabolic pathways like anaerobic respiration, using other electron acceptors like nitrate or sulfate or fermentation. Most pathogenic bacteria actually fall into this category, allowing them to survive in various body sites with differing oxygen levels. E. coli, Staphylococcus, and Streptococcus are common examples. Obligate or strict anaerobes. These bacteria can only grow in the complete absence of oxygen. In fact, oxygen is often toxic to them because they lack the enzymes like superoxide dismutase and catalase, which we'll mention later, needed to detoxify harmful oxygen byproducts formed during metabolism. They thrive in oxygen-depleted environments like deep tissues, puncture wounds, the gut, or canned foods. Examples include Clostridium tetani, causes tetanus, Clostridium botulinum causes botulism, and many bacteria involved in periodontal disease like Porphyromonas gingivalis. Microaerophiles. These bacteria require oxygen for growth, but they grow best at low oxygen concentrations, typically around 2–10%. Atmospheric oxygen is about 21%. Higher oxygen levels can actually inhibit their growth or be toxic. Examples include Campylobacter jejuni, a common cause of food poisoning, and Helicobacter pylori, associated with stomach ulcers. They often have oxygen-sensitive enzymes.
Wow, such diverse oxygen needs. What about pH? How acidic or alkaline the environment is? Does it matter much to bacteria?
pH is definitely important. Most bacteria prefer to grow in environments with a neutral pH, typically around 6.5 to 7.5. Optimal growth usually occurs around physiological pH, 7.2, 7.4, which is the pH of human blood and tissues. Bacteria that prefer neutral pH are called neutrophiles. However, some bacteria are adapted to extreme pH conditions. Acidophiles thrive in highly acidic environments, sometimes at pH values below 3. Examples include lactobacillus species involved in fermentation, like yogurt production, and also contributing to dental caries by producing acid in the mouth, or bacteria found in acidic mine drainage. Alkalophiles, on the other hand, grow optimally in alkaline environments, sometimes at pH 9 or higher. Vibrio cholerae, for instance, tolerates alkaline conditions well, which helps it survive passage through the stomach.
And the presence of water. We said it was essential, so drying must be bad for them.
Absolutely. Moisture is critical. Water is essential for all metabolic processes, acting as the solvent and participating in many reactions. Severe drying or desiccation is lethal for most vegetative, actively growing bacterial cells. This principle is used in food preservation methods like drying fruits or meats. However, this is another area where bacterial endospores demonstrate their incredible resilience. As we discussed, spores are highly resistant to desiccation and can survive in a dry state for very long periods, waiting for moisture to return. Light is another factor. Generally, darkness favors the growth of most non-photosynthetic bacteria. Ultraviolet, UV light, particularly short-wavelength UVC, is strongly bactericidal because it damages bacterial DNA, causing mutations and interfering with replication. This is why UV germicidal lamps are used for sterilizing surfaces, air, and water in labs and healthcare settings.
What about osmotic pressure, like salty or sugary environments?
Due to their rigid cell walls, bacteria are generally more tolerant to variations in osmotic pressure than animal cells are. The cell wall protects them from bursting in hypotonic solutions where water rushes in. However, they can still be inhibited or killed in extremely hypertonic environments. Very high solute concentrations like strong salt brines or sugar syrups. In these conditions, water rushes out of the cell, causing the cytoplasm to shrink away from the cell wall, plasmolysis, which disrupts metabolism and growth. This principle is used in preserving foods with high salt or sugar content. Bacteria that are adapted to high salt concentrations are called halophiles.
And finally, can physical forces harm them?
Yes, mechanical and sonic stresses can. Strong mechanical forces, like grinding or vigorous shaking with beads, can physically disrupt bacterial cells. High-frequency sound waves or sonication can also be used to break open bacterial cells in the lab by causing cavitation, formation, and collapse of tiny bubbles.
Okay, so temperature, oxygen, pH, water, light, osmotic pressure, physical stress, all these factors play a huge role. This really raises an important question. How does our understanding of these specific environmental preferences directly influence so many practical applications? Things like how we preserve food, refrigeration slows mesophiles, drying removes water, canning uses heat for thermophile spores, how we sterilize medical equipment, autoclaving uses high heat, some processes control oxygen, and maybe even how we try to target bacterial infections within the body, perhaps by manipulating their local environment. It's all connected. Finally, let's look under the hood at the sophisticated inner workings of bacteria, their engines, so to speak. How do they produce the energy they need to live and grow? How do they carry out all the complex chemical reactions of life? And crucially, how do they pass on their genetic blueprint to the next generation, sometimes with surprising variations that help them adapt?
Right. Let's talk about metabolism and genetics, the core processes of bacterial life. Bacterial metabolism encompasses all the chemical reactions that occur within the bacterial cell. These reactions are needed to obtain energy, to synthesize cellular components like proteins, lipids, nucleic acids, cell wall, and to grow and reproduce. These processes can be broadly divided into catabolism, breaking down molecules to release energy, and anabolism, building complex molecules which requires energy. And as we touched upon with oxygen requirements, these metabolic processes can be aerobic, using oxygen, or anaerobic, occurring without oxygen.
So how do they actually generate energy, the ATP currency they need?
Energy production is key. The main pathways bacteria use to extract energy from nutrients, like glucose are aerobic respiration. This is the most efficient pathway used by aerobes and facultative anaerobes when oxygen is present. It involves the complete breakdown of glucose through glycolysis, the Krebs cycle, ultimately using oxygen as the final electron acceptor in an electron transport chain located in the cell membrane. This generates a large amount of ATP, anaerobic respiration, used by some anaerobes and facultative anaerobes when oxygen is absent. It's similar to aerobic respiration in using glycolysis and an electron transport chain, but it uses an inorganic molecule other than oxygen as the final electron acceptor. Common examples include nitrate, NO3, sulfate, SO42, or carbonate, CO32. It generates less ATP than aerobic respiration, but more than fermentation. This is an anaerobic process used by many anaerobes and facultative anaerobes when respiration isn't possible. It involves only glycolysis to partially break down glucose, producing a small amount of ATP. The final electron acceptor is an organic molecule like pyruvate or a derivative. Fermentation results in various end products like lactic acid, ethanol, acetic acid, etc., which are often characteristic of different bacterial groups and are important in food production, yogurt, cheese, alcohol, and also in disease, acid production causing tissue damage. It's a fascinating detail related to oxygen metabolism. Oxygen, while essential for aerobes, can paradoxically be toxic because its use in respiration can generate harmful reactive oxygen species, ROS, like superoxide radicals O2, and hydrogen peroxide, H2O2. These can damage cellular components like DNA, proteins, and lipids.
So how do bacteria that live with oxygen survive these toxic byproducts?
They've evolved specific protective enzymes. Most organisms that live in the presence of oxygen produce enzymes like superoxide dismutase, SOD, which converts superoxide radicals into oxygen and hydrogen peroxide, and catalase, or peroxidase, which break down hydrogen peroxide into water and oxygen, catalase, or just water, peroxidase. Obligate anaerobes typically lack these protective enzymes, which is part of why oxygen is toxic to them. Testing for catalase production is actually a common diagnostic test in the microbiology lab. For example, Staphylococci are catalase positive, Streptococci are catalase negative.
And you mentioned carbohydrate metabolism plays a significant role in their interaction with us, even in our mouths, causing cavities.
Yes, carbohydrate metabolism is particularly critical in the etiology, causation of many diseases. A prime example is dental caries, or cavities. Bacteria living in dental plaque on our teeth, like Streptococcus mutans, ferment dietary carbohydrates, especially sugars like sucrose that we eat. This fermentation process produces acidic end products, primarily lactic acid. The accumulation of this acid locally on the tooth surface lowers the pH, and this acid directly demineralizes the hard tooth enamel, dissolving the calcium phosphate minerals, eventually leading to the formation of cavities. So bacterial metabolism of sugars is the direct cause. Many bacteria also produce characteristic enzymes that are incredibly useful both for their identification in the lab and often contribute directly to their pathogenicity, their ability to cause disease. For example, Staphylococcus aureus produces an enzyme called coagulase, which clots blood plasma. This is a key diagnostic test for differentiating it from less pathogenic Staphylococci, and the clotting might help the bacteria evade host defenses. Other examples include enzymes like collagenase and hyaluronidase, produced by some invasive bacteria which break down collagen and hyaluronic acid components of host connective tissue, helping the bacteria spread deeper into tissues. And as we mentioned, enzymes like galactomases, which degrade a lactome antibiotics like penicillin, are crucial for antibiotic resistance.
Okay, that covers how they generate energy and carry out reactions. Now, what about the core of their identity, their genetics? How do they store, replicate, and sometimes change their genetic blueprint, which allows them to adapt and evolve so incredibly quickly?
Bacterial genetics is a fascinating field, underpinning their remarkable adaptability. Let's start with the basics: DNA structure and replication. As we've established, the primary genetic material containing all the inherited characteristics is typically a single, large, supercoiled, circular chromosome made of double-stranded DNA located in the nucleoid region. DNA replication in bacteria is a highly accurate process, ensuring that each daughter cell gets a faithful copy of the chromosome. Semi-conservative, each new DNA molecule consists of one old strand and one newly synthesized strand, and usually bidirectional replication force proceed in opposite directions from a specific starting point called the origin of replication, or ORIX. The process is remarkably fast, keeping pace with their rapid cell division. And remember, bacteria are generally haploid, having just a single copy of their chromosome and thus a single copy of each essential gene. This means that the effects of a mutation, a change in the DNA sequence, are often immediately apparent in the phenotype, the observable characteristics of the bacterium, as there isn't a second gene copy to potentially mask the effect.
So mutations are one way they can change. How else does genetic variation arise in bacteria, allowing them to evolve?
Right. Genetic variation is the raw material for evolution and adaptation, and it can occur in bacteria through two main mechanisms. Mutation. This is a spontaneous random change in the DNA base sequence. Mutations can arise from errors during DNA replication or from damage caused by environmental factors like UV light or certain chemicals. While many mutations might be neutral or harmful, occasionally a mutation might confer an advantage, such as resistance to an antibiotic. These beneficial mutations can then be selected for, leading to adaptation of the population. Gene transfer, or horizontal gene transfer, HGT. This is a major driving force of bacterial evolution, allowing bacteria to acquire new genes or traits from other bacteria rather than just inheriting them vertically from their parent cell. This allows for much more rapid adaptation than relying solely on random mutation. There are three main mechanisms of HGT in bacteria. Transformation, where a bacterium takes up naked DNA fragments released from dead, lysed bacteria in the environment directly across its cell wall and membrane. Transduction, where bacterial genes are accidentally transferred from one bacterium to another by a bacteriophage, a virus that infects bacteria. The phage mistakenly packages some bacterial DNA into its viral particle and injects it into the next bacterium it infects. Conjugation. This involves direct cell-to-cell contact, usually mediated by a sex pilus, where genetic material, most commonly a plasmid, is transferred from a donor cell to a recipient cell. This is a very efficient way to spread plasmids carrying advantageous genes, like antibiotic resistance genes.
And plasmids, again, prove to be a really big deal here in HGT, don't they? Especially when it comes to the rapid spread of things like antibiotic resistance.
They absolutely are central to HGT, especially via conjugation. To reiterate their importance, plasmids are those smaller, usually circular, double-stranded DNA molecules that exist and replicate independently of the main chromosome. Crucially, as we said, plasmids often carry genes that confer properties like antibiotic resistance. For example, genes for alactomases, efflux pumps that pump antibiotics out, or enzymes that modify the antibiotic, or genes for the production of toxins or other virulence factors. Because plasmids can be easily transferred between bacteria through conjugation, they act as vehicles for rapidly disseminating these advantageous genes throughout a bacterial population, or even across different bacterial species.
Yeah.
A single plasmid carrying multiple resistance genes, an R plasmid, can quickly turn a previously susceptible bacterial population into a resistant one.
So what's really fascinating here, and frankly a critical challenge for us in modern medicine, is how bacterial genetics, particularly the role of plasmids and horizontal gene transfer, contributes directly to the alarmingly rapid evolution and global spread of antibiotic resistance. This is a major public health crisis, and it's a direct consequence of these intricate bacterial processes of adaptation and gene sharing. It's a real-world, high-stakes application of everything we've just discussed about bacterial physiology and genetics. Wow. You've just taken a really comprehensive deep dive into the microscopic world of bacteria. We've explored their incredible history, haven't we? From those first mind-bending observations by Leeuwenhoek to Koch's foundational postulates that finally allowed us to rigorously link specific microbes to specific diseases, we've unpacked their fundamental differences from our own eukaryotic cells, that prokaryotic simplicity versus eukaryotic complexity. We've glimpsed their diverse shapes, from cocci to bacilli to intricate spirals, and explored their intricate internal workings and external structures, including those amazing survival pods, the endospores that defy extreme conditions. And finally, we've really dug into their dynamic life cycles, that growth curve, their precise nutritional needs, how they masterfully adapt to almost every environmental challenge imaginable: temperature, oxygen, pH, and the truly fascinating world of their metabolism and genetics, including those game-changing plasmids.
Absolutely. So the next time you think about bacteria, maybe try to move beyond just seeing them as germs or, simple tiny organisms. There's so much more than that. Consider their ancient lineage. They've been on this planet for billions of years. Think about their omnipresence, thriving in every corner of our planet, from miles underground to high in the atmosphere, and of course, all over and inside us. And reflect on how their sophisticated adaptations, things we've talked about, like their unique cell wall architecture allowing for antibiotic targets, their diverse metabolic tricks, their clever genetic mechanisms like plasmids and horizontal gene transfer, and that incredible spore formation strategy have allowed them to not just survive, but thrive and evolve for eons. They are not just pathogens causing disease. They are absolutely essential components of every ecosystem, performing vital roles in nutrient cycling, like nitrogen fixation, decomposition, supporting plant growth, even aiding our own digestion and training our immune systems. And perhaps they even hold keys to future biotechnological innovations, new enzymes, new bioplastics, maybe even new medical treatments derived from their unique biochemistry.
That's a great point. They're architects and recyclers as much as they are sometimes adversaries. So a final provocative thought for you to ponder. What might the next big discovery about these tiny powerhouses be? How will further understanding of their intricate world, their communication, their community behavior, and biofilms, their vast unexplored genetic potential, change our understanding of life itself, and maybe even how we live? We really hope this deep dive has given you a fresh perspective on the invisible world that's constantly shaping our own. Keep exploring, keep questioning, and keep being curious.